Hi everyone. So this is part one of the blood vessels chapter. And so what we're going to start off with in this chapter is going over kind of an overview of blood vessels and kind of how they're arranged in the body. And then we'll talk in detail about the histology of a blood vessel. Okay, so let's do our overview really quick. So when blood vessels, they leave the heart, as they leave the heart and move further and further away, they become smaller and smaller. So the blood vessels that are leaving the heart would be the arteries, right? So the aorta, and then the vessels that branch off of that would be arteries. And then those would branch into smaller vessels called arterioles. Okay? And so then the arterioles branch into capillaries, all right, and capillary beds, which we're going to talk about in detail, all right? So these would be the smallest blood vessels, okay, would be our capillaries. And then from there, they start to get larger again as we move to venules and veins, which take blood back to the heart, all right? And so then eventually all of the veins would merge together into either the superior vena cava or the inferior vena cava as they enter the heart. All right, and one other thing to point out about this kind of progression through the different types of blood vessels, this is just the pathway blood would take, is that as we move from an artery, all right, to a vein, we decrease pressure. All right, so we have lower and lower pressure as we go through. So this would be a high pressure vessel. All right, and then the veins would be very low pressure, with essentially the right atrium of the heart having, no pressure at all. And so this is important because that pressure is what's going to keep blood moving through those blood vessels. And so the blood is going to be moving from high to low pressure. So you need that gradient of really high pressure in the arteries and low pressure in the veins to keep blood moving through those blood vessels. Okay? All right, and so with this picture is just kind of an example of what we're looking at. So with kind of the branching, just an illustration. So we would have maybe an artery right up here, and then that would branch to an arterial here, okay, and then that arterial would branch into a capillary bed. All right, so these capillaries we would see here, these would surround all of the tissues wherever gas exchange needs to occur. So you would have capillary beds in the tissues. All right, so there'll be capillary beds in the tissues because you need to exchange gas, provide oxygen to the tissues, take away carbon dioxide from the tissues. And then there would also be capillary beds in the lungs, all right, to do the opposite. So in the lungs, you would be adding oxygen to the blood and getting rid of that carbon dioxide waste product. Okay, so once gas exchange has occurred, all right, we would move then to a venule, all right, and a vein. Okay, and so we'll look at later when we talk about blood flow through the capillary beds, we'll talk about these kind of bands of smooth muscle that are going to help control blood moving into the capillary beds, all right? But for now, this is just the overview, okay, and how blood moves through the blood vessels. All right. Okay, and one other thing to point out on this picture is this kind of unique vessel that's illustrated down here. All right, this would be an arteriovenous anastomosis. All right, and so an anastomosis is just a connection between two different blood vessels. So this particular type of anastomosis, an arteriovenous anastomosis, connects an artery and a vein. All right, and so it's unique because you're connecting an artery to a vein without a capillary bed in between. Okay, so that's this arteriovenous anastomosis. And so we talked about an anastomosis before with the coronary circulation. All right, this is another example of a specific type. So you can have connections between arteries and veins without a capillary bed in between. Okay. All right. Now let's talk about histology. Okay. And so with the histology of a blood vessel, we'll talk in general what the kind of layers are, and then we'll go through and look at how the histology of an artery is different from the histology of a vein. Okay, so let's go through the general layers. All right, so when we look at this picture, this is, this is actually a picture of an artery, but we'll use it for illustration purposes, okay? And so we'll start on the outside and work our way in. All right, so the outermost layer, and we call these tunics or tunica. The outermost is the tunica externa. All right, so that kind of yellow layer on this picture, that would be the tunica externa. It's made of generally collagen and elastic fibers, right? So just connective tissue. And it'll give some structure to the blood vessel. All right, so that's the tunica externa. And then the middle layer, the tunica media here, that is made up of smooth muscle. All right, so smooth muscle and sometimes elastic fibers. And so depending on the blood vessel we're talking about will determine how much elastic tissue is in that tunica media. And so we'll see when we compare this between arteries and veins that the tunica media in an artery is going to be much thicker than in a vein. All right, and so that allows arteries the ability to change their diameter. All right, so change the diameter. All right, and so that is called vasoconstriction. Right, so if we make the blood vessels smaller and don't think I'm going to have enough room, I might, dilation. Yeah, there we go. All right, so we can vasoconstriction, constrict the blood vessel by contracting that smooth muscle. That will make the blood vessel smaller. Or vasodilation, if that smooth muscle contracts, it will make the diameter of the blood vessel larger. All right, so that's the tunica media, that middle layer. And then the innermost layer, the tunica intima, or sometimes I've seen it written tunica interna, right? Both are correct. The tunica intima, this is the layer of endothelium, like we talked about before with blood clotting, that endothelium on the inside of the blood vessels. Okay, so essentially this is the endothelium. So you'll have simple squamous cells, some connective tissue underneath it, and that is going to provide that smooth surface so platelets don't stick. All right, remember it secretes prostacyclin to prevent platelets from sticking. All right, and it is continuous with the endocardium of the heart. All right, so it's continuous with the endocardium, because you need that smooth surface because blood's going to be moving from the heart into these blood vessels. You need a very smooth transition. So this tunica intima, or the endothelium of the blood vessel, is continuous with the endocardium of the heart. All right, and so then one last thing to point out with these blood vessels, and it's not drawn in this picture, but large blood vessels have what's called vasa vasorum, okay? And so vasa vasorum is actually a blood supply within the blood vessel to supply the muscle and everything like that. Blood supply in large blood vessels. Okay, and so small blood vessels would not need this vasa vasorum because the nutrients from the blood moving through the vessel could just simply diffuse if it's not too thick of a wall in that blood vessel. All right, it could just diffuse to the cells that need those nutrients. But as the blood vessels get larger and you get this thicker wall, it's much more difficult for those nutrients and gases to just diffuse to the cells that need them. So you'll need kind of a little circulatory system running through the walls of these blood vessels. And so that's called vasa vasorum. So tiny blood vessels within a larger blood vessel. All right. So now let's be a little more specific. Let's talk about arteries and veins specifically. Okay. So with arteries, there's a few different types of arteries. All right. And so their histology is going to be slightly different, just based on their kind of function and their characteristics. All right, so our first type of artery is an elastic artery. And as the name will tell you, this type of artery would have a lot of elastic tissue in its walls. So all three of the tunics, the externa, media, and interna, would have elastic tissue. All right, and so that allows these vessels the ability to stretch and recoil very easily. Okay, and so an example of this would be the aorta, right, that branches directly off of the heart. All right, so it needs, because there's so much pressure with the blood leaving the heart, it needs the ability to stretch in response to that pressure and then recoil. Okay, so the aorta and its very immediate branches off of that heart would be these elastic arteries. All right. And so then our next type would be a muscular artery. All right, so with the muscular artery, they have a very thick tunica media. It's going to have some elastic fibers in there as well. And so these are going to be most of the named arteries. So the ones that you learn in lab, most of those, like the renal artery, the hepatic artery, the femoral artery, those are going to be these muscular arteries. So most of the ones with names that you have to learn in lab are going to be muscular arteries, and they're going to supply blood to specific organs. All right, so they supply specific organs. Okay, and so they have the ability to change their diameter, all right, vasoconstrict, more so than stretch like those elastic arteries that we talked about. So the smooth muscle in their wall allows them the ability to change their diameter. All right, and then lastly, we have the arterioles, okay? And so the wall of an arteriole is much simpler than our arteries that we were talking about. So in an arteriole, you essentially just have your endothelium and a layer of smooth muscle, all right? Which, because that is such a simple wall, that means that these blood vessels can change their diameter dramatically, right? There's not these thick layers of tissue, okay? It's just a very thin layer of smooth muscle with simple squamous cells just inside of it for that endothelium. So they can dramatically change diameter. And that's kind of their function, okay? And so what they do functionally is to determine blood flow into capillary beds. All right, so they allow, blood to enter. If they dilate and if they constrict, blood won't move into the capillary bed. All right, and you could kind of see that in the picture we were looking at here. All right, so if we have our arterioles here, all right, so if it constricts, right, and gets much more narrow, that'll let less blood enter that capillary bed. And if they dilate, more blood will be able to enter that capillary bed. Okay, and so then what we're illustrating here is how they can change their diameter. So this would be vasodilation, all right? So going from a smaller arteriole to a much larger one, and that would result in a decrease in blood pressure, okay? And then going from a much larger more dilated vessel to a small one would be vasoconstriction. All right. Okay, good. And so these arterioles, and we'll talk about this in a little bit with flow into the capillary beds. These arterioles are controlled by different signals. So there'll be chemical signals like the need of a certain, like the tissues in that capillary bed, if they need oxygen, if they're making a lot of carbon dioxide or lactic acid, that kind of thing. Those signals are what's going to stimulate these arterioles to dilate or constrict. All right, so we'll talk about that in a lot more detail and what those different signals are. Okay, so let's talk about capillaries now. All right, so we've done arteries and arterioles. Now we'll talk about capillaries. And so capillaries are very simple, and they need a very simple, very thin wall, because remember, in capillaries, this is where gas exchange is occurring. Okay, so you want the absolute thinnest possible barrier between the red blood cell in the middle carrying that oxygen and the tissues outside that need the oxygen. Okay, so very, very thin wall for capillaries. It's just made up of endothelium, which remember is simple squamous. All right, and the basement membrane underneath it. All right, so if hopefully you remember back to ANP1, the basement membrane is just like a very thin, sticky layer of glycoproteins on the surface of the cells, right? So they would be on the outer surface of the cells here. That's what this kind of clear structure is. All right, and that sticky surface just allows those cells, that endothelium, to adhere to the tissues around it. It kind of acts as, I always describe it as just like a layer of Velcro on the outside of the cells. Okay, so you just have simple squamous cells with some basement membrane on the outside. All right, and so in a capillary bed, the blood flow through the capillary bed is the slowest. All right, and so this allows for nutrient, we'll just say exchange, right? Because it could be nutrients or waste, okay? Whatever needs to be exchanged. And so just to clarify, right, to make sure you don't get these concepts mixed up. So the speed of blood flow, right, the speed of blood flow is not necessarily the same as blood pressure, because remember, pressures are highest in arteries and lowest in veins. So capillaries are kind of in the middle pressure-wise, but they have the slowest blood flow of all the vessels, arteries or veins included. Okay, and that's just because they're so narrow and so tiny. They're actually, the smallest capillaries are more narrow than an actual red blood cell. And so those red blood cells kind of have to like bend and fold over in order to pass through those capillaries. So that's why it's so much slower. And it has nothing to do with pressure and more so to do with their diameter, and they're so much more narrow. Okay? All right. And so because the blood's moving so slow and those capillaries are so narrow, the red blood cells end up passing through single file. Okay, and that is critically important so that nutrient, right, the waste and nutrient and gas exchange can occur. So that will allow maximum contact with that red blood vessel or red blood cell and the edge of that vessel. So we can have the red blood cell, right, pressed right up against the wall of this, capillary here. All right, so that makes it very easy for oxygen to be exchanged, right? It can give up its oxygen, it can take in carbon dioxide. So that's important for the function. Okay. And so most body cells, right, tissue cells in your body are located near capillaries, with the exception you learned with your tissues of cartilage, which doesn't have any blood vessels, it's avascular, epithelium, which doesn't have any blood vessels, the cornea and the lens in the eye, which also don't have any blood vessels. Okay, every other tissue, those cells, would be somewhere close to a capillary. All right, so there are 3 types of capillaries that we're going to talk about. Okay, and so these capillaries are going to be different based on kind of the cell itself and its structure and the amount of space allowed between cells. And so in capillaries, there are these, I'll kind of put it up here, whoops, with the pen. There are intercellular, so that means between cells, clefts, okay? And these intercellular clefts between the cells are kind of like gaps between cells, okay? And we'll see with some of the types of capillaries, they have more of these clefts than others do. All right, so that would be between those endothelial cells. All right, so there'd be like little gaps in here. Okay. Okay, so let's talk about the different types. So the first type is continuous. So this would be an unbroken layer of cells, very few of those intercellular clefts. All right. This is going to be the type of capillary we'll see in most tissues. All right, so this is that typical capillary bed that provides circulation to your tissues. Okay, and these continuous capillaries will have very few intercellular clefts, but there'll be some that'll allow for some solutes to be exchanged, small solutes. However, in the brain, right, there are no clefts whatsoever. All right, in the brain. And that's, if you remember, you have that blood-brain barrier, so you need very, very strict regulation of what is allowed to go from the blood to the brain. All right, so there's no clefts, so it's only diffusion that is allowing, things to be exchanged in the brain. Okay, so then after continuous, we have a fenestrated capillary. Okay. So a fenestrated capillary has pores or fenestrations, all right? And you can see that in the picture here. So it just has like these little holes through the actual endothelial cells themselves, so pores, all right? And so these blood vessels are good for filtration and absorption. Okay. And so the pores allow these capillaries to exchange larger molecules than a continuous capillary would. And so we find this type of capillary in the kidneys, all right, and in the small intestine. Okay, so the kidneys filter the blood to make urine, right? So you need, some larger gaps for that purpose. And in the small intestine, you have a lot of absorption of nutrients that needs to happen. So that would be necessary there. All right, and then lastly, we have our sinusoids. Okay, and so a sinusoid, you learned an example of a sinusoid already, all right? But a sinusoid is a capillary. It has lots of large gaps, right? Those clefts open up into these really large gaps. And you also have fenestrations, pores, within the cells. Okay, there's very few tight junctions between the cells in these sinusoids. All right. Okay. And so with the sinusoids, actually, I don't think we've even gotten to this. So with the sinusoids, one example of this would be in the liver. All right, and actually, we will talk about it when we get to the digestive system, right? You'll learn about those sinusoids. All right, so an example of a sinusoid is in the liver. And so it allows for movement of much larger molecules. Okay. All right, so now let's talk about blood flow through a capillary bed. All right, so capillary flow. Okay, so flow through a capillary bed is intermittent, okay, and we call that vasomotion. All right, so that's just the kind of like think of it as like a valve, all right, that allows blood to enter into the capillary bed, or it can shut off blood getting into the capillary bed. All right, that's vasomotion, kind of opening and closing of blood flow through those capillary beds. All right, and so you don't want blood going through every single capillary bed in your body at all times, right? That would be a very wasteful use of blood. All right, you want it to go to the tissues that need it at any particular time. All right, and then the ones that don't need it at a particular time won't get that blood flow through the capillary beds. Okay, and so this is controlled by these precapillary sphincters. All right, whoops. And so that would be these guys here, all right, and this guy here. Okay, so there are these bands of smooth muscle. And so they can constrict and cut off blood flow into that capillary, or they can dilate and relax and allow blood to enter that capillary. And so some of the signals that stimulate them to open, all right, would be these, okay? So they'll open and kind of relax, dilate in response to low oxygen. So these cells here, right, if they have a low level of oxygen, that is going to trigger these precapillary sphincters to open, all right? Because that's saying we need blood, we need the oxygen from the blood, okay? If there's also high carbon dioxide, so they're making a lot of CO2, they need to get rid of that waste product, that'll open up those sphincters, as well as a buildup of hydrogen ions and lactic acid, okay? So these are all waste products that when they build up, that will trigger the sphincters to open and send blood into that capillary bed. Okay, and so let's talk about just think about some examples of why you might need different, capillary flow through different tissues at different times. All right, so right after you eat a meal, the tissues that obviously are going to be needing blood the most, right, is going to be your digestive tract. So we're going to increase blood flow to the GI tract. That would make sense, right? Because that's what's working right now. It's digesting that food, it's doing a lot of absorption and all that kind of stuff. So you want to send the blood flow to there, all right? Especially if you've had a big meal, you're resting, you're sitting on the couch. Your skeletal muscle, for example, is not going to need a lot of blood flow at that time, right? It's not moving around a lot. So you're going to close off the capillary beds in your skeletal muscle, and you're going to send blood to the capillary beds in your intestines. Okay? So kind of counter to that, right? The opposite example would be if you're going running. All right? So if you're going running, you would increase blood flow to skeletal muscle, all right? And you would decrease and shut down blood flow to the GI tract. Okay, so it's just selecting what tissues need blood at a specific time. All right? Okay, and so then our last topic on this video is going to be talking about veins. Okay, and so with veins, let's kind of compare their structure a little bit. All right, so with veins, you can see in this picture of a vein, first of all, that this tunica media here is much thinner than in the artery we looked at before. All right, so it's going to be a thinner tunica media because it doesn't need that ability to constrict or relax. All right, so it's lower pressure, so it doesn't need that ability to change its diameter. All right, and then the tunica externa, on the other hand, is going to be a good bit thicker. All right, so it's going to have a thicker tunica externa than arteries. And again, that relates to the fact that there's not much pressure going through veins. All right, so this will help to keep the veins open, all right, when that pressure is low. So because there's not a lot of pressure with the blood moving through these veins, they need that kind of extra support to hold them open. And that's what that tunica externa is going to help to provide, keep them from collapsing. Okay? And so that tunica externa is going to have a lot of collagen fibers to kind of help give it structure and prevent collapse. Okay. And then another really important feature of veins that's unique is that they have valves. All right, so this would be a valve in this vein. So that valve is essentially a fold of the tunica interna, right? So this is a fold of tunica intima. Okay, so it's continuous with that endothelium, all right, which is important, so you make sure that platelets don't start sticking. Okay, and so it has kind of a semilunar valve structure, like the semilunar valves in the heart. So when blood is pushed up through this valve, all right, it can't go back down. It's kind of caught in these like little pouches. And so that's very similar to the way the semilunar valves in the heart work. All right, so it's going to keep blood moving towards the heart. All right, so this is what's going to keep blood moving towards the heart. Okay. All right. And so with veins, we have a specific type of vein that you have learned about before, or at least you've probably heard of, a sinus. We talked about the coronary sinus with the heart. So what exactly is a sinus? And so this is sinus referring to blood vessels, not sinus in the bones in your skull, right? That if you have the sniffles, right, you would think your sinuses are acting up. Okay, so this is a different type of sinus. And so a venous sinus is just a vein with no smooth muscle in its wall whatsoever. Okay, and so we learned the coronary sinus as an example of this. And you learned last semester about the superior sagittal sinus in the brain, right? Remember that goes between the two hemispheres of the brain. All right, so that was an example of a vein with no smooth muscle in its wall. Okay, so it's just a special type of vein. All right, and so let's talk about, because these veins are so low pressure, how, and they're fighting gravity most of the time to get blood back to the heart, right? Except for maybe with the head, where gravity will be bringing, venous blood down towards the heart. With your arms and your legs and your torso, these veins are going to be fighting gravity, and they don't have a lot of pressure to help them. So how does blood get back to the heart? All right, so some factors that help with venous return. Okay, so the first one is skeletal muscle milking, and this is essential. All right, so your skeletal muscle, let's imagine this vein has like a skeletal muscle, right, on either side of it. It's kind of embedded in there between them. It's all tucked in between your muscles in your legs, for example. And you just walk around, right? You're maybe doing some housework or something. You walk around the house. The muscle, when it contracts, it's just gonna squeeze in on this vein, and that is gonna squeeze blood up the vein towards your heart. So just the act of moving and using your muscles helps to move blood back to your heart. All right, and so that's one reason it's so dangerous with really long flights. If you're not moving for a long time, you should get up, walk around. If you're bedridden, or somebody maybe who's who's unconscious in a hospital, you want to move their limbs around to keep that blood moving. Okay, because remember, when blood pools, blood starts to clot, and that's not a good thing. Okay, so skeletal muscle milking. And then respiration actually helps with venous return. So specifically inspiration, right? So breathing in helps bring venous blood back towards your heart. Okay, and so what it does is it creates a negative pressure in the thoracic cavity. All right, so the way I always think of negative pressure is the idea of like when you're sucking through a straw. So when you suck on a straw, inside of that straw, you're creating a negative pressure. That negative pressure then draws the fluid up, that you want to drink. All right, and so that's kind of the idea of what's happening here. So we're creating a negative pressure in the thoracic cavity. So that's going to draw that venous blood up towards that thoracic cavity. All right, and then we talked about just a moment ago valves, okay? And so valves will prevent backflow of that blood. All right, so it prevents it from moving backwards. So then, the blood, as you have that skeletal muscle milking and, negative pressure pulling the blood towards the heart, once it moves past each valve, it can't go backwards. All right, so that helps too. Okay. Last topic, all right, and we're going to stop here, is just the idea of a portal system. So a portal system is a vein between two capillary beds. All right, and so there's an example of this, and you'll see this when we do the digestive system, where you have in your small intestine, right? So in the small intestine, you're gonna, eat food. It's gonna be absorbed. We're gonna absorb the nutrients from that food. So you would need lots of capillaries, all right, in that small intestine to help absorb all those nutrients from the food. The blood from those capillaries is gonna be passed on to what is called the hepatic portal vein. All right, so this is a vein, and then from there, that blood is then going to be sent to the liver. All right, and so the liver is going to contain a bunch of capillaries. Remember the sinusoids we just talked about. All right, and that is actually going to help to filter and sort out those nutrients. What needs to be stored, what needs to be, sent as a waste product, broken down, that kind of stuff. The liver does all that kind of organizing. All right, so there's a capillary bed in the small intestine, then you have the hepatic portal vein, and then there's a capillary bed in the liver. And so that portal vein in between, all right, that is part of the portal system. That's the portal part of its name, just means that it's sitting between two capillary beds. Okay? All right, so we're going to leave that here, and then we'll pick up and talk about blood pressure on the next video.